Hello everyone. Today I'm going to talk about the hemoglobin estimation, that is the practical we'll start from today. Okay. So I'll just give you an overview of what I'm going to talk about today. That is what is hemoglobin and what is the importance of estimation of hemoglobin. Then methods to estimate hemoglobin, SAHLI'S procedure to be followed, and then CYANMETHAEMOGLOBIN method, advantages and disadvantages of all the methods, and then I'll give a conclusion orally. So what is hemoglobin? Hemoglobin is a conjugated protein present in RBC which serves the function of transportation of oxygen and carbon dioxide between lungs and tissues. You know that hemoglobin is a part of the RBC. It is a globular protein, and in the center you have this hydrophobic molecule, and then in the periphery it is having a hydrophilic structure. So basically the structure is something like it can form a colloid type thing in the water, right? So that's the reason why it is associated with red blood cell. It is an integral part of it which is essential for carrying the oxygen molecule. As you know that each hemoglobin molecule is composed of 2 pairs of globin chains and 4 heme molecules, right? In the center you have this, in the heme structure you have this iron and having the 4 attached porphyrin structures, and then there is an additional pairing that is happening with the histidine, and also the additional bond is with the oxygen. So that's how the heme and the porphyrin ring and iron will form the heme. So basically it is forming a globular structure, all right? And then the 4 heme molecules and the 4 globin chains all together will form the hemoglobin molecule. Okay, so basically this the hemoglobin is can be a relaxed state or in a tense state, depending upon the hydrogen and the ionic bonds that are present. Okay, so if the hydrogen ionic bonds are ruptured, then they are in a relaxed state, and if they are present, they are tense, then it is in a tense state, right? If the hydrogen ionic bonds are ruptured, then it is in a relaxed state. So this helps in bonding of oxygen and then transportation of the oxygen to different tissues. So here we have the hemoglobin normal structure. You can see that there are 2 alpha chains and 2 beta chains here, right? Those are the globin chains and the iron molecule, that is the Fe2 plus and the porphyrin. Okay, so basically there are 4 heme groups here, and then the globin chains 2 alpha and 2 beta, that makes a HbA molecule. So basically we have this quaternary structure which is having the alpha beta kind of structure, right? So we have certain types of hemoglobin, right? In the adult we have this HbA type which is composed of the alpha and the beta. When we are talking about an embryonal hemoglobin, so usually we have when the embryo is formed, even then there is some nutrition that the embryo needs to actually make the organs start organification and all of that. So initially when you have this embryo, it has a hemoglobin known as GABA1 and GABA2 and Portland 1 and Portland 2. Just remember the names so that you know that there is a hemoglobin pertaining to the embryo, that is called the embryonic hemoglobin, as such like that of HbA or HbS in the sickle. You are giving it a name like in the embryonic hemoglobin, you are giving it a name like GABA1, GABA2 and Portland 1 and Portland 2. Okay, this is made of alpha chain and the epsilon chain. Basically, the globin chains are the ones which are making the major difference in the structure of the hemoglobin, by which we say that this hemoglobin is of this type. So the globin chains are important. So when you have this alpha and the epsilon hemoglobin, alpha and the epsilon chain, then you're having this embryonic hemoglobin. Then coming to the other one, see, after the embryonic hemoglobin, you have the fetus formed. In the fetus, the first 6 months, you have this HbF, that is a fetal hemoglobin, that is usually necessary for the mother's oxygen to be transported to the fetus. So that is HbF hemoglobin. Okay, Hb and F. So you have the embryonic hemoglobin, that's GABA1, GABA2, Portland 1, Portland 2. And in the fetal hemoglobin, you have the HbF. Then as the fetus grows, the HbF is replaced totally by the HbA, what we are talking now, that is the alpha and the beta globin chains, right? So basically the HbF is something which is having a gamma chain associated with the alpha chain. So in the embryonic hemoglobin, you have the epsilon chain, and in the fetal hemoglobin, you have the gamma chain. In the adult hemoglobin, you have the beta chain associated with the alpha, right? So there is another one where we see in something like thalassemia or something, you are finding HbA2 type of hemoglobin. In the HbA2 type of hemoglobin, you will find a little bit of HbF left. And then you find 2 alpha polypeptide chains associated with 2 delta polypeptide chains. So this is the structural difference what you see based on the globin chains. There is the categorization or the typing of the hemoglobins, right? So what are the normal hemoglobin levels? The normal hemoglobin is 70 to 24 grams per deciliter in the newborn. So in the newborn, if you're finding the cell counter report as when you ask for a CBC, you are having a cell counter report giving you 22 or something, it's normal, it is fine. But if you see the same in the adults, then you must think of some pathological abnormality or change. Then children 11.5 to 15.5 grams per deciliter, adults 13 to 17 grams per deciliter, females 12 to 16 grams per deciliter. So I've told you what is hemoglobin and importance of it. Then I told you about the categories or the types, the embryonic, fetal, adult, A2 type. And then there are some abnormal hemoglobins also. Before proceeding to what increases and decreases the hemoglobin, I would just like to tell you that there are some abnormal hemoglobins. As you have already finished the hematology chapter, you must be knowing that HbS associated with sickle cell anemia, right? So something like that, you have HbS, HbC. You know, there are some abnormal hemoglobins also which can be present in the body in different disease states. So remember the normal physiological types like embryonic, then fetal, then adult, then A2 type in something like thalassemia, and some patients can have A2 in some proportion with the A. You know, the proportions may vary with individual. Then we have some abnormal hemoglobins. The importance of estimation of hemoglobin is necessary because it is by hemoglobin that the oxygen gets carried to different tissues. Right? Without hemoglobin, you won't get necessary oxygen. So because of this oxygen carrying capacity of the hemoglobin, in the oxidized state, it is called as, when it is carrying oxygen, it is called as oxyhemoglobin. When it is not carrying oxygen, it is called as deoxyhemoglobin. And when it is carrying carbon dioxide, it is called as carbaminohemoglobin. And when it is carrying carbon monoxide, it is called as carboxyhemoglobin. So according to what is carried by the hemoglobin, either oxygen or carbon dioxide or carbon monoxide, we are giving it different names. That is, oxygen carrying hemoglobin is oxyhemoglobin. Deoxyhemoglobin when it doesn't carry oxygen. When it carries carbon dioxide, it is called as carbaminohemoglobin. When it is carrying carbon monoxide, then it is called as carboxyhemoglobin. You know that among the oxygen, carbon dioxide and carbon monoxide is having the higher affinity to the hemoglobin molecule. So if you keep a patient in a closed space with carbon monoxide, carbon dioxide and oxygen are already present in the air, but the hemoglobin will potentially choose the carbon monoxide and displace oxygen, carbon dioxide, right? So with this thing about the abnormal hemoglobins, type of categories and all that, we'll just go to increased, what happens if there is an increase in hemoglobin levels. So physiologically, newborns, I told you, can have 70 to 22 grams per deciliter of hemoglobin, so that's normal. Then higher altitudes, obviously there is hypoxic conditions, so in that we can find increased hemoglobin because it is trying to compensate the hypoxia after exercise, and diurnal, that is more in the afternoon. Then pathological. Pathological is polycythemia, wherein there is increased in the stacked red blood cells that you can see when you kind of sediment. Then the burns, then condition of severe dehydration, obviously it is like a relative increase in the hemoglobin levels because the volume of the fluid in the body is low. Then cyanotic heart disease. Now, decreased hemoglobin levels are seen in absolutely all types of anemia and in pregnancy due to hemodilution because the volume overload state is pregnancy, right, physiologically. So whenever there is volume, increased volume, the RBC tend to or seem to be decreased, but they are not decreased. So we have this decrease in the hemoglobin level due to hemodilution. So what is the significance? To know whether the person is anemic or not, use for calculation of red cell indices, which will help in typing of anemias. So when we are talking about the estimation of hemoglobin, see, for any normal screening purpose, you do a hemoglobin. Hemoglobin gives the nutrition status of the person. Whenever you want to subject the person for the surgery or something, you try to do a normal a complete blood cell count so that you need to see that the patient can undergo the surgery or not, something like that. So for the screening purpose, to check the nourishment of the patient, then any ill-nourished person or malnourished person you're finding, or if there is any breathing difficulty, then you will also think in terms of hemoglobin estimation. So methods of hemoglobin estimation are by estimation of the color intensity of the thing, then measuring the specific gravity of the hemoglobin and measuring the iron content of the hemoglobin, the blood, oxygen combining capacity of the blood. So usually we are doing the color intensity comparison. The specific gravity, iron content, oxygen combining capacity are not done in routine. So this is the most common principle. So what do we do in the colorimetric method? We kind of match the color of the HB with the solution with a suitable standard, either by direct vision or a photoelectric calorimeter. So you kind of make a solution from the hemoglobin of the patient's blood, and then you are trying to compare this color with the standard. So what are the types of these colorimetric methods? So we have Tallquist method, SAHLI'S acid HEMATIN method, alkaline HEMATIN method, Halden method, photoelectric CYANMETHAEMOGLOBIN method. So we have 5 methods. So coming to Tallquist method, we have to compare the color of a drop of blood which we put on the blotting paper, and then we have a colored paper in the booklet, and basically we just compare. So when we are mass screening some villages or something, then it is useful, or school camps, then Tallquist method is useful. Coming to the other method, SAHLI'S acid HEMATIN method, this is the procedure what we do in the exam and also in our labs. So this is the SAHLI'S acid HEMATIN method. We will talk about SAHLI'S in detail. Okay. So before that, I'll talk about the alkaline HEMATIN method. HB is converted to alkaline hematin by addition of N/10 sodium hydroxide solution. Okay, what is the problem here is that the fetal hemoglobin is not usually converted, but the carboxy, methemoglobin, hemoglobin, all are converted. So hence this method is used to detect the presence of fetal hemoglobin. Okay, so alkaline hematin method is used to detect the presence of fetal hemoglobin. Halden method, here HB is exposed to carbon monoxide, so carboxy hemoglobin is formed. Basically, the Halden method is very accurate, but why are we not doing that is that carbon monoxide is not good, right? So that's the reason why it may be potentially dangerous to be used in the lab. So that's the reason why the Halden method, though accurate, is not usually used. Then photoelectric CYANMETHAEMOGLOBIN method, it is a perfect and accurate method. We'll discuss about CYANMETHAEMOGLOBIN method also in detail. So we have known the 5 colorimetric methods, that is a Taliquest method, acid hematin, or the SAHLI'S method, alkaline hematin method used for HbF, Halden method which uses carbon monoxide, that's the reason though accurate we're not using, for electric CYANMETHAEMOGLOBIN method. All these are the colorimetric methods which are used preferentially over the other methods. Then coming to the second one, that is specific gravity. A drop of blood is allowed to fall into series of copper sulfate solutions of known specific gravities, and marking whether the drop rises to the surface or sinks to the bottom. If the blood drop sinks, its specific gravity is greater than that of the solution. This is used in blood banks for screening blood donors. So once we conduct a blood donation camp, okay, so blood bank requires some packets of blood to be stored in the blood bank, right? So once we call for example, generous people like the GVP students will come and donate a lot of blood. They want to donate. So can everybody donate blood? No. Then everybody can be checked for CBC and HB? That is time taking. You can't ask the person to sit all through, right? So what do we do is take the person who is a voluntary donor, you take just a little bit of blood, a drop of blood. Then you pour into a bowl where you have this copper sulfate solution. Okay, this copper sulfate solutions are put in a series, and then you have known specific gravities of each. For example, you label a bowl as A. A is having this specific gravity, B this specific gravity, C this specific gravity, and then A you put a drop of blood, then it is just sinking. Then the specific gravity is greater than that of the solution. And if it is not, then put in B. Then if it is not, put in C, something like that. Okay. Then this is used in blood banks, I told you. So this is this is a method which is used in the blood banks. The third one is chemical method. This is an indirect method wherein the HB level is done by estimating the iron content of the whole blood. So for usually the heme is the one which is having iron in the center, the porphyrin rings, 4 porphyrin rings and the histidine, and the other one bond is for the oxygen, I told you, so forming a globular structure. So in this test, the iron is first separated from the hemoglobin by an acid, then complexed with a reagent to develop a color, which is measured photometrically. It is complex, difficult and time consuming, but this method is accurate. So the first method is the colorimetric method. In that we have seen 5 types: the Taliquest, SAHLI'S, alkaline hematin method, and then we have seen that there is this Halden method and photoelectric CYANMETHAEMOGLOBIN method. The second type, specific gravity for mass reading of blood banks. The third one, that is a chemical method, which is an indirect method. And then in this, we have the iron measuring iron, we measure the iron. That's how we get the hemoglobin estimation. So this iron has to be separated from the heme, which is bound in the heme to the globin chains and the heme. The iron is separated, and then this iron which is separated is mixed with the reagent, and that forms a color. Okay, it's a very difficult method, but it is accurate. Something like Halden method, which uses the carbon monoxide, is accurate, but we cannot do have the carbon monoxide. Then the other one is a gasometric method. It is a complicated method, but accurate method for standardized variation, which is seen in the hemoglobinometers. Okay, it is also an indirect method. Amount of hemoglobin is estimated by measuring the amount of oxygen it is absorbing. So, according to the amount of hemoglobin that is present is estimated by using the amount of oxygen that is absorbed in the. So usually we are doing a color comparison, a specific gravity comparison, ion estimation, and the fourth one is the measurement of the gas that is present associated with the hemoglobin, that is usually the oxygen. So you measure the oxygen and then you quantify the hemoglobin which is present. So these are the four methods. So the other one, what very easy method is the hemoglobin estimated by the CYANMETHAEMOGLOBIN method, that is done automatically by a cell counter. It is like a machine. You do not require any procedure to be done. You just take the blood and then you just give it a shot into the automated cell counter. It has a straw-like thing. It just inserts inside and then it sucks the blood. Then you get a result. It's a very easy process, and the automated cell counters inherently have the CYANMETHAEMOGLOBIN method. So I told you this is a SAHLI'S acid HEMATIN is the one which we do in the our labs, and this is also for your exam. So that is a part of the colorimetric method. So what is the principle here? The acid HEMATIN is formed by adding N/10 HCl to a sample of the blood. So this color of the acid HEMATIN is measured visually by comparing with the standard which is given in the SAHLI'S HEMATIN method. You have this components, right? In that you get this comparator. So you should always remember, whenever you do an experiment, you have to remember the principle. Okay, formation of acid HEMATIN by N/10 HCl to a sample of blood. That should be your answer, right? So we have these things which are the materials. This is the comparator. This has the color which we can match to it. Then there is a hemoglobinometer tube that can be inserted here. Then you have this hemoglobinometer pipette, the stirrer, and then there is a marker here, and then 20 microliter mark is here in the hemoglobinometer pipette. The same thing. So we have this distilled water, all the requirements, the sterile cotton, glass rod, the hemoglobinometer pipette, I told you the 20 microliter mark, the comparator, the graduated tube, 90% ethyl alcohol, that is spirit, N/10 HCl, and the sterile needle. So basically what you do, you take N/10 HCl to the 20% or 2 gram mark in the hemoglobinometer tube, then add the 0.02 ml of blood to it by using the hemoglobin pipette. So basically first put N/10 HCl into this. So you're putting N/10 HCl into this tube, okay, up to 20 mark. Then you take this hemoglobinometer pipette, you puncture the blood with this sterile needle, put a ethyl alcohol swab, clean it, clean the surface, then puncture your skin surface. Then you have to put this hemoglobinometer pipette near the blood drop which is coming out. Then the blood drop has to be sucked in until this mark, and then you have to make it like this, and then you keep until 10 minutes. Okay, because you are trying to convert it. Okay, after putting it inside. Then dilute it to match with the standard color by adding distilled water drop by drop. Stir it with stirrer to ensure proper mixing. The percentage of hemoglobin can be directly read from the scale on the hemoglobinometer. So I don't think there is any confusion. You will have a spirit on your finger, or if you're directly getting blood from somewhere, it's fine. Otherwise, you take the person's finger, then put a spirit in it, spirit cotton, sterile cotton. You'll rub it, then puncture it with the needle. Then you take the hemoglobin pipette and then up to this mark, and then you kind of 0.02 ml that accounts for, and then you put uh, the blood of the hemoglobinometer pipette into this graduated diluting tube, which you will keep in the comparator box. After adding N by 10 HCl, after 20 mark, you add this 0.02 ml of blood inside it. Then you wait for 10 minutes, you stir it, wait for 10 minutes because you want the formation of acid hematin. Then once after 10 minutes, what you do is you add drop by drop distilled water. Then once you add drop by drop distilled water, you will try to match the color of the graduated tube to the comparator's color. Then once you get the color, you will measure what kind of mark is it matching with. Then you will get the hemoglobin percentage. So what are the advantages? It is simple method, cheap method, easy method, does not require much training. Disadvantages: estimates only oxy and reduced hemoglobin. Serum, bilirubin, lipids, proteins can influence the final color. Visual matching sometimes, visual matching can be a subjective error. I can say that it can be 14 grams and you can say that it is 16 according to our visual correction of the color. Color standards may fade, that is the comparator thing. The maximum color development usually takes about 40 minutes, that we usually don't wait. Errors in equipment, sample dilution, color matching and reading. So that can be present. So again, I'm repeating this. Take N by 10 HCl until the 20% mark here in this graduated tube. Then take blood until 0.02 ml. There is this mark here. Take blood until that mark. Put that inside. Stir it. Wait for 10 minutes. Then add drop by drop distilled water. Match the color with the comparator standard. Right. So that is about the SAHLI'S acid hematic method. You have to remember the principle, and the most gold standard method for estimation of hemoglobin is a CYANMETHAEMOGLOBIN method that is also used in the automated cell counters now. It is the best method, so you have to remember the principle of CYANMETHAEMOGLOBIN method also, that is a potassium ferricyanide oxidizes hemoglobin into methemoglobin. Then the cyanide ions of the potassium cyanide combine with the methemoglobin to form cyanmethemoglobin, the color of which is measured by means of photoelectric calorimeter at 540 nanometers. So there is, what we are using here is a potassium ferricyanide that is forming methemoglobin from the hemoglobin of the patient's blood, what you are taking. So this methemoglobin again will be reacting with the cyanide ions of the potassium cyanide. Then it is forming a cyanmethemoglobin. So potassium ferricyanide oxidizes hemoglobin. Methemoglobin is formed. Methemoglobin reacts with cyanide ions, then forms cyanmethemoglobin. And there you get a color, and then it is having, you can read this color by photoelectric method. What you are having is a Drapkin solution. See, many people can ask you what is the best method, what are the advantages, types of hemoglobin, what are abnormal hemoglobin, what is the principle of SAHLI'S method, what is the principle of CYANMETHAEMOGLOBIN method, and also they can ask you about what are the ingredients in the Drapkin solution. You have to just say potassium ferricyanide, potassium cyanide, dihydrogen, potassium phosphate, and distilled water. No doubt potassium ferricyanide, potassium cyanide, dihydrogen potassium phosphate, and distilled water, calorimeter, and the blood sample. Right. So how much blood you are taking? 20 microliter. Added to 5 ml of napkin solution, allowed to stand at room temperature for 3 minutes. Then there is a color that is developing, and then you compare it with the calorimeter. So how much amount of blood? 20 microliter. Napkin solution, how much? 5 ml. How much time? 3 minutes. So compared with the SAHLI'S method, this is less time consuming, and then you are having a faster about method, right? So this is the materials you have to remember: the ferricyanide, potassium cyanide, dihydrogen potassium phosphate, and distilled water. I'm repeating again and again because this can be asked in your exam. What is the components of the napkin solution? So advantage is most accurate method. It measures all the hemoglobins except the sulfhemoglobin. The color intensity again is measured by the photoelectric calorimeter at 540 nanometers, as we told you. So no visual error, no subjective error like that of the SAHLI'S. Simple method, short time, I told you 3 minutes. Stable color red compound will develop, so can measure at our convenient time. So from 3 minutes at any time up to within the 24 hours thing we can measure. So that's the reason why this is the best method. There are some disadvantages. So what is it? The method will not measure self-hemoglobin. Napkin solution obviously is having potassium ferricyanide and potassium cyanide. So it is highly poisonous, should not be pipetted by mouth. Plasma proteins, increased lipids, WBC will interfere with color development that can be seen even in the SAHLI'S method. Requires special equipment like the photoelectric calorimeter and requires training to do the test, unlike the SAHLI'S acid HEMATIN method. So that is about the HB estimation. You can have questions regarding the structure of the hemoglobin, the types of the hemoglobin, until what age there is fetal hemoglobin, and what are the components or what are the globin chains that are giving the structure of the hemoglobin, what are the abnormal hemoglobins. People, the examiner can ask you about the classification of anemias, then he can ask about the hemolytic anemias, then the confirmatory thing or the best method that is used for the hemoglobin estimation, that is the CYANMETHAEMOGLOBIN method, the components of the napkin solution, then the SAHLI'S acid HEMATIN method. That principle you are absolutely, you cannot afford to forget the principle and the procedure of the SAHLI'S acid HEMATIN method, right? Thank you. So I'll be joining you again for the urine examination. In the meantime, you will have peripherals, where and other classes going on with the practicals, right? Thank you.